next speaker is Charles Guan from Caltech. And his talk today is going to be on decoding finger movements from human posterior parietal cortex. So Charles, welcome. Go ahead and share your screen.
Great, thank you, David. So give me 1 second to pull up speaker notes. All right, I'm Charles Guan, a PhD candidate in Richard Andersen's lab at Caltech. Today I'm going to be presenting our work on decoding finger movements for neuroprosthetic control. In able-bodied people, motor commands from the brain descend the spinal cord to the effector, such as an arm or hand. Disruptions in that signal pathway, such as a spinal cord injury, however, can result in paralysis. Even after paralysis, however, attempted movements can still be decoded, enabling paralyzed participants to control a computer interface like a tablet, or one day control a prosthetic hand. Hand control is a large part of everyday life and a top priority for people with tetraplegia, so our study here focused on hand movements, and specifically on individual fingers. Many previous hand BCIs have been limited to a few grasp templates, while humans have pretty unique control over finger individuation, which is useful for a lot of day-to-day function. So we're hoping to build up from individual fingers to more flexible hand BCIs eventually. To record neural activity, we implanted a 96-channel electrode array into the left posterior parietal cortex of 2 tetraplegic participants. Using fMRI mapping, the implant for one participant was placed at the junction of the postcentral and intraparietal sulci, which I've shown in the orange circle. And this region is thought to be involved in the planning of grasp movements. I've shaded the larger PBC in green on the right, which is involved in high-level motor planning, reach and grasps, and eye movements, among other things. We also work with another participant with implants in both PBC and primary motor cortex, which I've shaded in blue. I'll focus specifically on results from participant 1 because we have a little more data in this talk, but I'm happy to answer questions about both participants as well. And I've focused specifically on PBC results as less is known about individual finger movements in this brain area. So our behavioral task, over 10 recording sessions, we instructed the participants to attempt to press individual fingers of their right hand, including a no-go condition. And here are the letter abbreviations I'm using in this presentation. I'll note that because our participants are paralyzed, they are attempting to move their fingers, but they can't actually move them. We compared firing rates of single neurons during movement execution against the no-go condition. And here's one particularly selective neuron. In the top plot, each dot is a single trial. The x-axis is the condition, so the different fingers, and the y-axis is the firing rate. The bottom plot shows the distribution of the mean firing rate differences against the no-go condition. And units are normalized by the variances. So you can see that this neuron increases its firing rate significantly during the thumb and index movements, but not for the other fingers. Now, if we extend this to using the full recorded population of about 100 neurons, and we use a linear classifier, we can then classify movements solely from neural activity. And this is offline, so not with the participant in the loop. But this is a confusion matrix showing which conditions were classified. And here, higher values along the diagonal are better. We achieved a cross-validated accuracy of 86% and chances 17%. So using the same idea, we then mapped neural activity to finger movements in session so our participant can control a brain-computer interface. So each trial, the participant taps attempts to tap a finger, and then their attempted movement is now decoded. And using only a short calibration period for the classifier, the participant is able to use that to accurately control individual fingers of the BCI. And here's a confusion matrix on the left showing that the online control accuracy averaged 86%. And this is higher than any previously published work that we know of. This is also the first known result showing that PPC activity has information about all 5 individual fingers as well. So we're excited to see this. These finger representations were robust across contexts, and they can be used in a variety of environments, including to move a virtual reality avatar hand, which I'm showing in the video. And in this demo video, our participant is controlling the individual fingers of the VR hand to acquire the green gems simply by thinking about moving their fingers. Okay, so given that our participant had been paralyzed for many years and thus does not move her hands, we were surprised to find that her attempted movements still resulted in distinct neural patterns that we could decode. So this led us to two natural follow-ups. On the decoding side, we asked whether we would be able to decode additional types of movements aside from those 5 finger presses of the right hand. On the neuroscience side, we asked and we're curious how these fingers representations are organized, given that she had not used her hands for about 10 years at this point. And so I'll start with some discussion on the second point. So our consistent neural patterns are underlying how our participant is able to perform their BCI task well. And we wondered whether our participant had learned these finger representations or whether they'd been, whether she had been paralyzed, lost the representations, and then relearned them to perform the task, or whether they had been preserved after paralysis from when she was able-bodied before. To investigate this question, we used representational similarity analysis. We quantified the representational structure by taking the fire rates during each movement condition. So I'm showing here an example of the index and the pinky finger, and we're binning the firing rates during the actual movement or attempted movement, and calculating a cross-validated distance by using trials across the different repetitions of each finger. This gives us one element of a pairwise distance, and this is one element of the representational dissimilarity matrix. And if we combine over all the pairs, this gives us the full RDM. We distinguish between two models. First, if the neural representations were preserved, we would expect the finger representation to match the neural structure previously identified in able-bodied people in panel B. And this structure has been shown to follow the kinematics of natural movement. If the neural representations were instead learned to control a BCI, we would expect the finger representation to follow the optimal statistics of the task. And because the movements were independent in this BCI task, this would lead to an independent representational structure on panel C. And looking at the data, so this is the RDM for our participant averaged over 10 sessions. You'll see that the finger representation strongly matched the able-bodied data. You can see this by visually comparing D and B. But if you want a more intuitive visualization of what the structure looks like, here's a 2D projection using multidimensional scaling. You'll see that the thumb is neurally distinct and the middle ring and pinky fingers are more similar. And the ellipses here are confidence intervals over the 10 sessions. If we want to more rigorously quantify the match between our results and the models, we can use an unbiased cosine similarity. And in this violin plot, you can see that our results match the able-bodied model much more strongly than the task optimal model. So when I described the task optimal model, I also mentioned learning, though, and perhaps the participant had learned to produce the specific organization. If so, we would expect to see this learning modify the representational structure with usage. So we also checked that the RDMs were consistent across sessions. So here we split into the early session and the late session, and you can see visually that they're similar. And we also wondered whether there's learning within sessions. So splitting between run blocks of the same session into early and late, we also didn't see a consistent change between across time for that. So we don't think the participant had learned their way into generating this finger structure. So to summarize this section, we developed motor skills throughout our lifetime and particularly in our childhood. We asked what happens when our lived experiences fundamentally change, such as a severe injury. First, cortical reorganization could have erased those existing finger representations. Yet in the first section, we showed that we could still accurately decode finger movements. So there's information about finger movements still there. Second, the participant could relearn to operate a BCI, optimizing neural activity for the task. Or third, the motor representations could be preserved after injury. And our evidence from our tetraplegic participants suggests that this final hypothesis is correct, that motor representations are preserved after injury. So going back to the two directions I mentioned, I touched briefly on the neuroscience follow-up of the first section. And next I'll touch on some decoding extensions, specifically decoding additional movements from PPC. Specifically, we looked at decoding individual finger movements of both the left and right hands. And typically, cerebral hemispheres are thought to control the contralateral side of the body. We've only implanted in the left hemisphere. However, being able to classify both same and across sides would decrease the number of implants tetraplegic people would need in the future for BCI usage. And here's the confusion matrix for offline cross-validated decoding. And you can see that there's reasonable decoding of fingers of both sides. So the cross-validated accuracy is 69%. But this decrease is somewhat expected because we have more classes, and so the chance accuracy is also lower than 10%. If we look at the finger organization, thanks. If we look at the finger organization again, this might tell us something about the structure. And so here's the average dissimilarity matrix over those 10 sessions. So the top rows are left hand and the bottom rows are right hand. And each row now corresponds to a finger of each hand. And as with all RDMs, we see that there are 0 values on the diagonal because each thing has 0 distance to itself. However, it's also pretty clear that the distances are small on the anti-diagonal. So these are corresponding fingers of the left and right hand. So the left thumb and the right thumb are the anti-diagonal, or the right pinky matching with the left pinky. And this suggests that the representations between hands are somewhat shared. So if we zoom in on those values and compare matching fingers across hands, which are labeled blue, and matching and values between mismatching fingers across hands, which are labeled orange here, we can quantify and test that the distances are indeed smaller between matching fingers. So on the right plot, each circle is one element of the dissimilarity matrix, but we've aggregated across 10 sessions. So that's why you see that n equals 50 instead of 200. And you can see that the pairwise distance is on average 1 distance unit less than for matching fingers than for mismatching fingers. And we're only using the top right quadrant here because these matrices are symmetric, so it's the same as the bottom left quadrant as well. So these results show that left and right fingers are decodable, yet they share representations in this recording. So one open question that we're still trying to understand better is, why would the brain choose to do this in computational reasons? And this seems to be a subset of a recurring pattern that our lab has called partially mixed selectivity, where variables have a mixed but hierarchical coding. And there are many reasonable explanations, but this is still an open question, and we'd love to hear your ideas. So in summary, we decoded finger movements from PPC activity. The corresponding finger organization suggests that motor representations are preserved after paralysis, and we found shared but discriminable representations across hands. And there are some interesting future directions that would be to study. So more complex hand gestures and potentially continuous finger control. So with that, I'd like to thank you all for listening. I'd like to thank my lab and our funding agencies for their support. And most of all, I'd like to thank our participants who make our research possible. There's much more discussion I couldn't get to here, so if you're interested in learning more, you can check out our preprint on bioRxiv with this QR code. It discusses the first 2 sections of this talk, and we'll also release code and data and a follow-up preprint expanding on section 3, so if you're interested, feel free to subscribe with the second QR code. And thanks for listening. I'm happy to take any questions.
Thank you. Thank you so much, Charles. That was amazing. We have a lot of questions. I have tons of questions myself, but want to give priority to the people from the audience. So Kyle is asking, why did you choose PPC and not motor cortex or premotor cortex, for example?
Yeah, good question. This is somewhat of an opportunistic thing in that there are other studies going in our labs and there are limited areas to implant. So but we do have another participant implanted in motor cortex and PPC, a different part of PPC, and we find similar results in motor cortex. And talking to a few other groups that do similar studies with motor cortex implants, we do find similar, this is for the preserved motor representations part, they find similar results in their tetraplegic participants with motor cortex implants as well.
Awesome. Another question from the chat. Did you try motor synergies?
I'm assuming this is in reference to the preserved motor representations part. So I guess it is a little difficult to maybe tease out some of these in single finger movements. And I think it would be interesting to do this with more complex hand movements. So some of this work is inspired by a Diedrichsen paper, where they do also multiple fingers at the same time. And some of the models, the model that we use is borrowed from that for able-bodied movement. And they are comparing against either essentially muscle synergy models or kinematics. And it's difficult to tease those out in just doing individual finger movements, but that is definitely something we'd like to look more into.
Awesome, thank you. One more question that I have is like from the very first graph that you showed, like it looks like that the thumb and the index fingers have the biggest like separation across all the 5 fingers. Do you think that is related to the range of motions that we can do with those 2 fingers compared to the other 3?
Yeah, I think so. That's what it appears to me, at least, that we individuate and use our thumbs and most independently from the other fingers. I think our index maybe a little more so, and the middle ring and pinky fingers tend to be used more together. And so this calls into mind what I think someone was mentioning about motor synergies. This also calls to mind maybe there are biomechanical constraints, but it's interesting that we see this in neural activity as well, and preserved even after our participants haven't been using their hands for many years.
Awesome. All right. Thank you. Thank you so much for the presentation.